great pleasure to introduce Dr. Schulman. Paul Schulman is an associate professor in psychology, which most of you know. This study actually started in a seminar in psychology where the initial question was, how come people die of drug overdoses don't actually take more of the drug than usual? How come when they die of a drug overdose, they're not actually taking more of the drug than usual? Carly, come in, don't worry about it, join us, have coffee, have bagels, we're all friends here. The term overdose is actually a misnomer. The general idea is that drug tolerance involves a homeostatic response, and that response is learned, something I didn't know, but we're going to learn about it, aren't we? So you're going to be telling us about this during your talk.
Yes.
Okay? So why don't you just take it away and tell us all about it.
Okay. This is a quote from somebody I like, and it relates, I guess, to biology. If any of a multitude of physiological variables varies, I don't like that. Outside a narrow range for any length of time, we get sick and then we die. The term wisdom of the body is about mechanisms by which we stay within these narrow parameters, and learning is one of the factors that gives the body its wisdom. Okay? And I did start this, I did do this study as part of a seminar initially. And, my training, that's the word, is in perception, is in vision. And so when I started doing seminars here, I did them on visual perception. And people said at the beginning, in the middle, and at the end of the seminar, what? What is this about? So I figured I would go find some other topic, and the topic I did find that I thought would be, sexy and compelling was that question. How come people die from so-called drug overdoses when it seems that they don't take drug overdoses? Okay? And from that, I got into quote. Here's what I'm going to do, is I'm going to explain, I guess, a bit of the history of this general area, and then I'm going to talk about 3 experiments that I didn't do on drugs, and then I will talk about the experiment that I am doing, that we are doing, and it's not done. So it'll be the coda at the end. So it's halfway done, I suppose. But, and I'm going to summarize, I guess now is a good time to summarize all of the experiments by this little anecdote, which is partially true, as all my anecdotes are. They're never entirely true. They're mostly lies. But this one is partially true. And when I was a child, at the end of our block, there was a fence, really a wall. And all the kids would walk on the wall and balance on this wall. And it was very narrow and fairly long. And so my brother, who's, an evil genius, certainly evil, would just gently tap my legs. And initially I would fall over onto the grass. So he was on the sidewalk here, grass here, wall here. So he would gently tap my leg, and because he knew that any idiot could bowl someone over on this narrow walkway. So the point was not to overpower somebody, but to make somebody look like an idiot. So he would just give me a gentle tap, and initially I would fall on the grass. And then as an organism, I quickly realized that as he got close to me, he was going to push me. And so I kind of leaned into his push, and I would balance it or compensate for the push. And so, because he would give me a bigger push. And so I would lean more into the push. So I would make a response. Okay? But being, and so he would actually eventually push me quite hard, but I could compensate for it, and everything worked out. But then he realized, because he is an evil genius, he realized that if he stood next to me and pushed between my legs, missed me, that I would fall on the sidewalk. And I did. So I fell on the sidewalk, tore my pants, tore my knee up, ran home, and then my brother, I guess, meandered home. And when he got home, my mother said to him, "What did you do to him?" And he said, "I didn't touch him." And that is my recollection. I doubt that it's really true. And then he decided that he would sort of induce a friend of ours to push me. So the friend wouldn't have knocked me over, but he just gently pushed me, and I fell over on the grass. Okay? So this whole story, as silly as it is, really encapsulates all the experiments that I'm going to talk about, or nearly all the experiments that I'm going to talk about. So my brother, you could think of as a stimulus, or the occasion, I guess, for during which I was pushed. His push is a stimulus. My push back, my compensation, is a response. And when the friend pushed me, that's very much like, that's the same stimulus as what my brother did, but it's not compensated for because I didn't anticipate it. Okay? So with this little story, and I suppose some experiments, I intend to explain drug tolerance and drug overdoses. We'll see how that goes. Okay, so I'm going to talk just a bit about the history of this stuff. And I see this in terms of homeostasis, that is in terms of a term invented by a physiologist named Walter Cannon. Okay? But the field was started in the mid-1800s by this biologist by the name of Claude Bernard, who I read was an iconic, like Einstein or like Stephen Hawking or somebody, was that era's iconic scientist. So this is a big deal, I guess. Anyway, so what Bernard did is, everybody must have known that when you're cold, you shiver and you put on a sweater, right? You have to know that. And when you're hot, you sweat and take the sweater off. And what this guy did is he actually discovered a mechanism in the blood that regulated blood temperature. So he figured out that when it's cold, the blood vessels constrict and pull away from the skin, the surface of the skin, and thereby sort of insulate you better. And when it's warm, they do the reverse. They dilate, expand, and move towards the surface of the skin. Okay? And he thought of that as a regulatory mechanism. And there could be, dozens and dozens of others. And the other thing he discovered, and these were, rather than discovering just the commonsensical things that we might, where if you're hot, you sweat, he also discovered that the liver generates glucose. From raw materials, I guess carbohydrates. But it generates glucose, and that could also be part of a regulatory mechanism to maintain the blood sugar level. Okay? And a person, Walter Cannon, and this is a famous quote in this sort of limited famous quote area. All the vital mechanisms, however varied they may be, have only one object, that of preserving constant the conditions of life in the inner environment. Right? If the environment, if your guts are too cold, you can't move your muscles, and your heart will essentially stop, as I guess we saw in the movie Titanic. And if you're too hot, the reverse will happen. The blood will, you just can't survive. But anyway, he made this a generalization for all of functioning. Okay? And Walter Cannon, maybe 70 years later, an American physiologist, maybe the premier physiologist, I don't know, of the 1920s and 30s. Yes? I don't know. Anyway, he created this experiment about homeostasis, about hunger. And this is a very simple experiment, but it's nice. Here's what he did, is his idea was that hunger pangs, hunger pains, are the stimulus to eating. Okay? And when your stomach is empty, it has spasms. Okay? And when it has spasms, it triggers hunger. It triggers the sensation, the subjective sensation of hunger. I guess this is before television. It triggers the sensation of hunger. So here's what he did. Is he had his subjects swallow a gastric balloon. He filled the balloon up inside the stomach, so you can kind of see that this long tube, this guy actually swallowed it. I guess they hired sword swallowers. I'm not sure exactly. I really have trouble taking an aspirin. But anyway, they swallow this balloon, they fill it with air, and this device transduces it so that when the stomach has a spasm, the air is actually sent up. The volume of the stomach is sort of reduced, and the air would be pushed up the neck, both necks, and is recorded on this graph paper. The other thing that the person did is, whenever he felt the feeling of hunger, whenever he felt hungry, he would push this little button, and that would be recorded over here. And then around his stomach there was another sort of gauge that would measure whether or not he actually was squeezing his abdominal muscles, okay? Because you didn't want to confuse that. And here's what he found, is that when the stomach did have a contraction, when it had a sort of spasm, which we could now call a hunger pang, the person was more likely to say he felt hungry. Oh, so that's interesting. When the thing is, it's sort of too easy. But when the thing is empty, it has a spasm, and the person feels hungry. Oh, it doesn't matter if that's too easy, because it's a simple, nice model. Okay? When there's a tissue deficit, the body detects it and tries to restore it. Okay? And I guess if there's a tissue surplus, the body would detect that. Sort of a thermostat. The body would detect that and cease eating, or cease drinking, or urinate, or sweat, to remove it, to remove the surplus. Another sort of, I guess this guy is a neurophysiologist, but he was interested in behavior, and he had the same general idea. Kurt Richter, he's not a psychologist, but he's close. And here's the sort of thing he did. It's the same general idea. He would remove glands, and if the gland controlled some aspect of behavior, he would check to see if the behavior, if the animal would compensate for it. So, in this case, somehow the thyroid gland helps rats regulate body heat. So if he removed the thyroid and just watched these critters, they would tend to build larger nests, better insulation, in order to maintain their body temperature. And if you would give them the injections of the thyroid hormone, they would stop doing it. They wouldn't build nests. And he would also transplant thyroids into them, or bits of the thyroid into them, and that would also regulate their nest-building behavior. Same thing with the adrenal gland. It helps control salt metabolism. So you would remove the damn thing, and the animal would drink salt solution like mad. Had a real craving for salt. And in one case study that I read a while ago, there was this little boy who had a real craving for salt, an intense craving for salt. So intense that his parents would put the salt up on high shelves. The kid would climb the shelves. His parents would lock the cabinets. The kid would break them open. I don't know if he did that, but he would try. Anyway, his craving was so intense that they took him to a hospital, Johns Hopkins, I think it was, and the hospital gave him a normal hospital diet, and he died because he had an adrenal insufficiency of some sort. Okay? So the idea is that if there's a tissue deficit, you do stuff to compensate for it. If there's a surplus, then you'll do other things hormonally, internally, and maybe behaviorally to reduce the surplus. Okay? And all of these guys think of this as all these fellas think of this as reflexive. That is, it doesn't involve any thought. Okay? So these are quotations. I put the emphasis on all of them. So homeostatic regulators act automatically. Skeletal muscles and diaphragms are under control of the cerebral cortex. Their functions in the regulation of temperature, blah, 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 are managed low in the brainstem, which is to say reflexively. And here's another quote that I like. It's desirable to remove from physiology terms having psychological and botanical implications. And I guess that's what he means by botanical. Psychological, it's just, the psychological crap that we study in psychology, which he didn't like so much. And this says the same sort of thing. The idea is that it's reflexive, it's automatic, and it responds to tissue needs. Okay? But, so if it's reflexive, you might expect that Pavlovian conditioning might have some role to play in this. Right? Because Pavlov taught us, when I discuss Pavlov, people in my classes say, so what? It's about spit. Who cares? And it is, I guess, about spit. And my experiment is about spit, saliva. But Pavlov really did show that you could trigger reflexive responses, reflex-like responses. So in principle, if those compensatory responses, the homeostatic responses, a term that Cannon invented, if those homeostatic responses are reflexive, then in principle, learning should have a role, could have a role. But, and here's how, here's how, this is in color, but this is the sort of iconic experiment, right? He would sound a tone, you can't see that here, and then give the dog food. The dog would salivate when he ate the food, and the amount of saliva would be recorded here. Initially, the amount of initially in this sort of horrible way that these people used to do stuff is they used to cement a test tube to the he would divert the salivary gland outside the cheek and cement a test tube to the animal's jaw. But then he developed a more humane, if that's the word, more accurate, that's the word, technique. Okay? So he'd sound a tone, the animal would eat. When he ate, he would salivate, and he would record that. Then a tone. A few seconds later, he'd give him some food, the thing would salivate, blah, blah, blah. And then he would sound the tone alone. and the animal would salivate. So he did produce a sort of reflex, a salivary reflex. And what's interesting is that the reflex that he produced was the same as the biological, the natural reflex. So the conditioned reflex is the same, looks like the same, as the natural reflex. Okay? Because salivation to the tone looked the same as salivation to food in the mouth. Okay, so that's nice. But here's the odd thing. I guess it's of some historical interest to people who teach the history of psychology, if those poor souls exist. These two guys were friends. And when Pavlov and Cannon were friends, here they are together, photoshopped. They were not photoshopped. They were friends, and when Pavlov came to this country, he would stay with the Cannons. So they were really good friends as well as colleagues. They were both, famous biologists, physiologists. But they really didn't seem to have any effect on one another. It's odd, because Pavlov was interested in digestion and won the Nobel Prize for studies, biological studies, purely biological studies of digestion. And the other guy was also interested in digestion, among other reflexes, among other regulatory systems. But these guys had no intellectual effect on one another. And I don't know why. But here's a guess. And since I've been reading about this, because I thought I had to explain this, for Cannon's stuff to work, the response, I guess the learned response, would have to oppose the physiological response to regulate it, right? So if you put, if there's some sort of disturbance, sort of like my brother pushing me, if there's some sort of disturbance in the environment, you give an animal a drug, you make the animal cold. According to Pavlov, the learned response simulates, is similar to the physiological response. Okay? So salivation to food, a physiological response, seems the same, or virtually the same, as salivation to the bell. Okay? But in order for it to work for Cannon, they have to be opposed. One has to compensate for the other. Okay? And I think the other reason that, though in the literature that I've read, it doesn't say this, but this is the real reason. The other reason is that Pavlov argued that the conditioned stimulus, the neutral stimulus, like the bell, has to precede the trigger for the reflex. Oh, come on in.
I don't want to interrupt your thoughts.
Oh, that's okay. Whatever was I saying before this terrible interruption? So, the idea was that the neutral stimulus, the sort of nonsensical, the tone or the light or whatever, must precede the trigger for the reflex by just a few seconds. And when it came to eating and other stuff, it takes a longer period of time. So, I mean, Pavlov showed pretty clearly that the greater the distance between these two stimuli, the less likely and the less likely that it was that learning would take place, and the weaker the learning would be. But, like, in the late 1950s, a psychologist by the name of John Garcia, who is a really interesting guy, did this experiment where he poisoned rats by giving them an overdose of X-rays, and he fed them. So he would give them, let's say, saccharin water. He would give them an overdose of X-rays and simultaneously sweetened water. And then 2 days later, he would offer them sweetened or regular water, and they wouldn't eat the sweetened water. So if you give them the overdose of X-rays, they seem to associate it with taste, but the nausea that comes from the overdose follows by 20 minutes, an hour, or even several hours later. Okay, so it looks like for some biological stimuli, some naturally occurring stimuli, you can get associations that take place over a long span of time. And I guess because of the way Pavlov chose his model, it looks like he did it to make it as sort of arbitrary as possible. The tone has no relationship to food. So it seemed as arbitrary as possible because it seems like, it seems like it makes the best case. If you chose smell or taste, it seems like they could be hardwired to begin with. So he created a sort of artificial system. Anyway, in the 1970s, there were hints hints all along. But in the 1970s, a guy by the name of Shepard Siegel, a regular New Yorker, did experiments on insulin. Okay, so and I'm going to discuss 3 experiments that he did because I really like them. So he did experiments on insulin, and basically the experiments were very simple. He would give them insulin in one room, and insulin lowers blood sugar, right? And he would give them several doses of insulin, and then he would switch the, he would give them several doses of insulin, and then he would give them a placebo. Okay, so here's the basic experiment. He would give them the drug in a distinctive room, and it could be in the colony room, which would have an unpleasant smell. Maybe they wouldn't notice, or in another room, but the room was the key. And come on in, Mary. Anyway, so here's what he would do: is he would give them. First, there would be a baseline period, so which was 7 trials, and he would give them an injection during the baseline period, and then he would snip off a little bit of their tail, like a millimeter of their tail, and suck out a little bit of blood and test it for glucose. Okay, so he would do it in this sort of environment. So he did this. This was done. The baseline thing was done for 7 trials, but I guess when an animal is nervous or afraid, the liver, which is a sort of, I guess, reservoir of glucose, releases it so that the thing can fight or run away. But in this case, he couldn't do either. So in order to make him no longer nervous, there were 7 trials, but the last 3 trials became the baseline. So he would measure the amount of blood sugar on the last 3 trials. Okay, on the last 3 baseline trials. And then he gave him 4 trials of insulin training. And it would work like this. I think I have a picture of these guys. They would put them in, the baseline training would take place, in fact the whole thing would take place in these restraining tubes. This guy, I guess, is commiserating with this, what looks like a bunch of cotton balls, but is a white rat. Okay? They would give them the injections in one of these holes, and their tail would stick out another hole, and they could cut it off with a carving knife. You've never seen such a sight in your life. Okay, so they would do this for 4 trials, and then on the 5th trial, they would give him a placebo injection. Okay? Now, if you recall the story that I told at the beginning, my brother would push me and push me and push me, and I initially would fall over, but then after a while, I learned how to resist it. But then he pushed between my legs, and I fell over the other way, in principle. And that's like giving an animal a placebo. Okay? So this guy gave the animal a placebo, and these were the results. This explains it. This is the test conditions. This is a placebo test. There are several ways of doing it. I won't go into it. This is a placebo test. If you do it in the insulin-associated room, the room where you got the insulin, then the animal should produce a compensatory response, the way I did on the wall. And if you give them insulin, then the compensatory response should push one way, the drug should push the other way, and they should cancel to some extent. But if you give them a placebo test, which is what happened here, then if it's in the drug-associated room, you don't get the drug, but you should be able to see the conditioned response, naked of the drug, without the drug. Okay? So you should be able to see the compensatory response. And here's the way he set this up. He had lots and lots and lots of animals. I forget how many, but 7 in each condition. One set of animals was, the dashed lines are animals that were given saline solution throughout this thing. Okay? But the way it was set up is that one set of animals, I think it was 7, I forget, was given the injection of insulin and had their blood extracted after 5 minutes, one after 10 minutes, one after 20 minutes, one after 30 minutes, one subset after 20, 30, another subset after 40, up to 90. So these are different animals. It's not really the course of time with one animal. So we can see that the insulin effect seems to maximize around 30, 45 minutes or so. Okay? And then the graph up here, oh, and incidentally, this is the drop in blood glucose, right? I don't know if you can see these minus signs. But this is the percentage of baseline drop in blood glucose. And then he gives them an insulin, a placebo injection, and he got the reverse response. Okay? That is, their blood glucose went up. Okay, so it does look like if insulin lowers the blood glucose and the response is compensatory, by giving them a placebo, you can see the compensation. Okay? So in a way, it really is like my brother pushing me but missing on purpose, and me falling over towards him. So all you saw was my response. You didn't see his push. Okay? In the next slide, what he did was after 20 minutes, he repeated the experiment. But looking for what I guess is called a dose-response curve. So he would give one set of animals nothing, a placebo, one set of animals a low dose of insulin, a moderate dose, and a high dose for a separate set. And then he would extract their blood after 20 minutes, because if you don't remember, but it looked like the conditioned response peaked at 20 minutes. So this is the conditioned response, the learned response. It looked like it's compensatory, which would make Cannon happy, but it looked like for some reason it peaked after 20 minutes. So all of these took place after 20 minutes, and you can see that as you increase the amount of insulin that you gave to them for the first 4 trials, their blood sugar goes down and down and down, and the conditioned response, the response to the placebo, is the reverse. So it does look like there's a compensatory response. The animal is pushing back. Or, since I guess I have to relate it to the beginning, trying to maintain homeostasis. The insulin disturbs homeostasis, it lowers blood sugar. The animal is doing I don't know if it's ever conscious, but it's doing what it can to balance it, to compensate for it. It's producing a compensatory or a drug-negative response. Okay? And this next experiment was on morphine, because really no one cares about insulin, I guess. It's set up in a similar way. There were two rooms. One was the colony room, and one was some other room. The colony room stank. It was full of dirty rats or whatever. The colony room smells, and it was colder, if I recall, and it had a different level of, it was noisier. The other room was just different, okay? So in one room, in the colony room, the way it was set up was that you would be given an injection of morphine in one room. And 30 minutes later, your pain sensitivity would be measured. So some of the animals got their injection in the colony room, and in the colony room, they were measured, their pain sensitivity was measured with an analgesiometer, which is a fancy term for a device that pinches your paw. And what the animal is free to pull his paw away. Here I am. It's free to pull, it looks sort of like a stapler. So it puts pressure on his paw, and when he pulls his paw away, it measures the last amount of pressure. Okay? So if you got morphine, and somebody is putting pressure on your paw, you don't give a damn. So if he's putting pressure on your paw, it takes a long time. It reduces the pain. It takes a long time for you to pull it away. Okay? So half of them were trained in the colony room with the analgesiometer, and this does sound like Clue. The other half were trained in another room with a hot plate. And a hot plate is, as it sounds, they put the animal's paw on the plate, and they count the amount of time it takes for him to pull it off and lick it. So the longer he keeps it on, the more the morphine is working. The more powerful the morphine is. Okay? So there were 8, so half of the animals were trained in the colony room with the analgesiometer, and the others in the library with the pipe. The others in the other room with the hot plate. Okay? So that was the training. But for testing, they were tested in both rooms. So they got 8 trials of training in this room or in this room. But for testing, randomly arranged, they got tested in this room and in this room. Okay, with the analgesiometer or the hot plate. Okay, so let's just think about this. If you develop tolerance to the drug, you should pull your hand away from the hot plate faster. You should You should pull your paw away from the hot plate faster, and you should pull it away from the analgesiometer at a lower pressure. You should be more sensitive to pain. Okay? Now, this shows that the animals develop tolerance to the pain, because their pain measurements for half of them took place all along. And this is the analgesiometer, and these are grams of pressure. This is the control group, and they don't seem to change much, but this is the morphine group, and during training, and you can see that at the beginning they don't give a damn, but after a while there's really no difference between them. They develop tolerance to the morphine. There's no difference between them and the other fellows, the control group. And this is the paw lick latency. It's not as pretty, but the open circles is the control group that always got saline solution, and this one is the morphine group. Okay? So they develop tolerance. Now, according to this theory, if you develop tolerance, what you're doing is you're learning to produce a compensatory response. That is, you're somehow internally increasing sensitivity to pain. the drug reduces sensitivity to pain, you're increasing sensitivity to pain, you're developing tolerance to the drug. And the argument here is that the tolerance it's not a biological response, that's basically fixed. It's a learned compensatory response. Okay? So these were the results. If the animal was trained, acquired the tolerance in the hot plate room, he removed his and tested in the hot plate room. Okay, he was trained and tested in the hot plate room. This is training. This is testing. He removed his paw after 12 seconds. If he was trained in the analgesiometer room and then tested in the hot plated room, it took him twice as long to remove his paw from the hot plate. Okay? In a similar way, if he was trained in the analgesiometer room and tested in the analgesiometer room, it took 113 on average grams of pressure. But if he was trained in the other room and tested in the analgesiometer room, it took more than twice as much pressure. So since the drug-taking histories of these animals are identical, and actually their response, everything is identical. The only difference is if they associate the room with the drug. Okay? So these fellas associate the alternative room with the drug, and if they're tested in that room, they develop tolerance. These guys, if they develop tolerance, it's a little bit less. It's a lot less.
That's a question. Is it the room or the type of pain?
It's the room.
Because they're also getting a different type of pain in the other room. Is that?
That's a good question, and I left out a detail.
Oh.
And for half of these guys, the hot plate didn't work. They were just put in the room. And for half of these guys, the analgesiometer didn't work. And the numbers that I gave you average them because there was no difference between them. So what you could say is, well, if you put your hand on the hot plate, it takes a certain amount of skill, I guess, to remove it. To remove it. But half of these guys didn't have any practice. So it's not the device. It looks like it's the room. And it's not the drug because they were both given the same amount. It looks like the drug, the tolerance is associated with the room. Yeah,
The dosage remained the same?
Yeah. Yeah.
They didn't increase the dosage?
No, same throughout. You want them to increase the dosage? In the next experiment, they do. Okay, but the argument here is that if you associate the room, but if you associate the room with the drug, you produce responses that mitigate the effect of the drug. Okay? So that's the argument. And if you get the drug in another room, then you kind of blindside it. It's like having a friend knock you off the wall. You're not ready to be knocked off, to be pushed, and you don't produce the response that would, the compensatory response, and you get knocked off the wall. Okay? So in the next experiment, we knock them off the wall, or he does. And to my mind, this is the most dramatic experiment ever. And really maybe the best experiment in the history of psychology. It's certainly dramatic. Okay? So here's the basic idea, is that when people die from so-called overdoses, their lungs somehow swell, and they, I guess, basically suffocate. Okay? Their respiration is depressed, and their lungs swell, and they can't breathe. Okay? But the main point, I guess, which I mentioned earlier, is that they don't take more. Real people, it's hard to measure this because real people who die of real drugs don't do it in a neat way, and they tend not to document it. But overdose victims tend to be experienced users. They're not new users. So I guess they must know what they're doing. And on autopsy, overdose victims have no more heroin, I guess it's other drugs as well, but I've only read about heroin. No more heroin in their bloodstream than people who are heroin addicted, but die from gunshot wounds and strangling, I guess, from other means. Okay? So it doesn't look like it's the amount of drug. And it's not an extraordinary amount of the drug. Okay? Of course, this just says you'll never know. Because it's, it's a mess, right? Because they're living real lives, and they're not keeping track of everything. And they're not an experiment. Okay? But could it be that it's a loss of tolerance because you take the drugs under unusual circumstances? So could it be that, like my friend pushed me when he had never pushed me before, and I fell onto the lawn because I wasn't prepared to be pushed. He didn't push me hard, but I wasn't prepared to be pushed. I hadn't anticipated that I would be pushed, and I just fell over. Okay? And that's basically the argument here. So here's what Siegel did. He gave rats increasing doses of heroin. This involves real heroin. And there were 15 trials. They were every other day. So they would, on Monday they'd get a heroin dose in the colony room, and then the next day they'd get a placebo. Then the next day, heroin, placebo, blah, 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 blah. Okay? So one room is associated with the drug, and one room is associated with the placebo. And for half the animals, it's this room that's associated with the drug, and for the other half, it's the other room that's associated with the drug. So it would be switched. And then there was also a control group that was given an increasing dose of placebo, saline solution, under the same circumstances. Okay? So now we have two heroin groups, both of them really treated the same. We will have in a second two heroin groups. Both of them treated the same. They're both given increasing doses of heroin, and then there's a control group given increasing doses of saline solution, which shouldn't do anything, right? And it looks sort of like this when you glom together the two rooms. So this is the training. Is that what it says? It's maybe a little hard to see. This says that they got the drug in one environment, and then they split them for the test. Okay? For the test, where they really did get a lot of heroin, for the test, they either got it in the drug-associated room or in the placebo-associated room. Okay? They were trained in one room, and they got it either in the same room or a different room. And then there was also another group that got saline solution under the same circumstances throughout the whole damn thing. And then on the final day, and it was the final day for many of them, there were 28 control animals that got saline, 28, and all but one, I guess, died from the overdose. For those who got it in the different room, like 2/3 of them died. And for those who got the drug in the same room, right, it's the same room, the drug-associated room, only about 1/3 of them died. So since the drug-taking histories of the two drug groups was identical, it looks like the only difference is the room. Okay? So it looks like they learned to predict the effects of the drug based on whatever information is available. If I always get the drug in the bathroom, if I always take the drug in the bathroom, then I associate the bathroom with getting the drug. If I always take the drug with friends at a party, I associate that. So he actually did do a case study, sort of people who survived drug overdoses. And most of them took the drug under unusual circumstances. Okay? So they had Michael Barron, my friend, push me gently, and I fell over on the other side. So under unusual circumstances. So you could ask, well, why should it be the drug? Why should it be a room? Can't it be anything that predicts the drug? Can't it be anything that predicts the effect of a disturbance internally? Anything. And I guess you could say, well, if you eat something sweet, that's going to predict that there's going to be sugar in your bloodstream a few minutes later, 20 minutes later. Because that's a perfect predictor, except for saccharin and stuff like that. If you eat something that tastes sour, I guess that means that you'll have to digest acids in a short period of time, however that's done. I'll depend on Carly to tell me. If you eat fatty foods that taste of umami, whatever, then I guess the body will prepare itself to digest, in principle, to digest fatty foods, fat. Okay? So there's no reason why it has to be a room. A room is just a convenience. In real life, it's your brother, or it's the stuff you eat. There are better predictors, often. So the study that I did, and I did it with students here, and they are Rachel Hickman, David Franklin, and Brianna James. What? Brianna? No, not you yet. And they helped plan the study a year ago. And now we're doing it with Bianca Johnson, Francesca Palladino, and Shannon Daly. And they'll be doing this soon. There's no reason, and it's not, sad to say, it's not that original. It really is a replication of something that this Paul Rozin did. And the idea is this, is that, coffee is a drug, or caffeine is a drug. And people drink the stuff to get the drug to become alert or whatever. It's a little hard to measure alertness and probably expensive. It would take time. But one of the effects of caffeine is it makes you salivate. So that's what I measured. That's what we measured. And that's what we'll measure. It makes you salivate. Okay? So it suggests a natural experiment. If you compare coffee drinkers to non-coffee drinkers, coffee drinkers take caffeine all the time, and should have learned to mitigate the effects of the attentional effects, whatever the other effects are, and the salivary effects, all the effects of the drug. Because coffee has a characteristic taste which predicts those effects. Okay? So it could be Pavlovian conditioning. Those people who are not coffee drinkers, defined as by some survey where we asked them if where they said they rarely or never drink coffee, then they shouldn't associate the taste with the effects, the physiological effects, and they should not have learned the response to control them, to mitigate them, the compensatory response. Okay? So that's basically what we did. We gave everybody 8 ounces of decaffeinated coffee, Folgers extra something, and then on 2 occasions, randomly determined, and then we added caffeine. We added 200 milligrams of caffeine to the coffee on one occasion. Okay, and we may be doing that again here. We added 200 milligrams of caffeine to the coffee on one occasion. They could prepare it however they liked, however they were accustomed to preparing it, because that could be part of the clue. And we recorded saliva before they drank on 2 occasions. We always recorded it for 5-minute periods. And so we recorded it for 10 minutes before and then for 45 minutes after they drank. Okay? And we gave them 15 minutes, 10 minutes to drink, and then 5 minutes to clear their mouth of liquid. Okay? And we recorded it using these dental cottons, right, which we're all familiar with. And this was suggested to me by Paul Rozin. And all we did was weigh these things in this little baggie, which I got from a drug dealer. No, I didn't. And weigh them in these little baggies. Before we, I did go to Puff and Stuff, but I didn't like their bags. I really did. Anyway, I went to that Joanne's Hobby or something, I don't know. Anyway, so we weighed them in the bag before, and then they put them in their mouth for 5 minutes. We sealed the bag, and we weighed them again. Okay? And so these were the results. Red is coffee drinkers, blue is non-coffee drinkers. So let's look at the red. This is at 5 minutes after, 10 minutes after, 15, 20 after they drank the coffee. Okay? And so you can see that and the solid line is caffeinated coffee, the dashed line is decaf. So there's really no overlap, though there are only 16 subjects. But there really is no overlap. But this is not a surprise, I suppose. Caffeine makes you salivate, and no caffeine, I guess, doesn't, or makes you salivate less. Coffee, of course, is bitter, and that might make you salivate also. And the blue is the control group, so solid means they got caffeine, and dashed means they didn't. It's hard to figure out what all this means, but the next graph makes it simpler, I think. Maybe too simple. This one is these guys. Blue is non-coffee drinkers, red is coffee drinkers, and this is the baseline. So everything is compared to the baseline, as was the previous graph. And this is coffee drinkers getting caffeine, non-coffee drinkers getting caffeine. And this is non-coffee drinkers getting decaf, and this is non-coffee drinkers, coffee drinkers getting decaf. Okay? And this is with respect to the baseline. So the first 10 minutes we recorded the amount of saliva, and this is with respect to that. So it looks like they're producing a compensatory response, though we're only halfway done.
Were those significant differences?
No.
Oh, they weren't? Okay.
They're not, but I have high hopes.
Okay.
We also measured other stuff, but I haven't looked at that yet. So I guess the upshot of this is that what is the upshot of this? The upshot of this is that classical conditioning is far, far more general than anyone would ever have thought. It's not about spit, even though this is. It's not about saliva. It's about predicting what's going to happen next and producing responses that mitigate those effects. I guess someone who studies cybernetics would say, or at least to the extent that I understand it, if the temperature goes up, the thermostat goes off, it goes down, the thermostat comes back on, so it controls in that way. That's analogous to a tissue need for us, but it looks like we can predict when we're going to have, we, rats and us, can predict when we're going to have a tissue need and can produce the proper compensatory response. And I think it's very, very, very general. And it may not even involve a tissue need, but I think it's very general that you could even say that all of psychology, Joanne wouldn't say this, but she might. You could say that all of psychology is really predicting what's going to happen next and producing a response. And just to tell you how general it is, I think I just have another slide or two. People have done stuff on cold water swimming. If you throw an animal in cold water repeatedly, they'll be cold, but pretty soon they produce responses that reduce the effect of the cold water. And there is this one guy who is the world's champion, if that's the right word. Maybe it's just that he's stupid. He's the world champion cold water swimmer. What he does is he swims in Arctic rivers and lakes in Norway. So the water temperature is 32 degrees, or if there's salt water, it's less. Okay? And a typical person can survive for 5 minutes before their heart stops and their muscles stop, and he so far has been able to swim Lewis Gordon Pugh, Google him, P-U-G-H. He's so far been able to swim almost a kilometer, and it took him 19 minutes. We would be dead, just like Leonardo DiCaprio, Titanic. We would be dead. But he has somehow managed to compensate for that. And there's a clue in one of his videos, because he says that before he jumps in the water, he runs a temperature. His temperature is 102. Okay, which says to me he's doing this. People have done stuff with the opposite saunas. With eating, of course, I think it probably works. If you eat something sweet, you predict that blood sugar is going to rise, and you release insulin to mitigate it. And it even works in perception. And this is something called the McCulloch effect. It's a wonderful, wonderful, wonderful effect discovered by an undergraduate at one point. Named Celeste McCulloch. And here's what it is. If you look at this thing for a period of time, longer than I'm going to let you look at it. If you look at a red thing, you get an after image that's greenish. If you look at a blue thing, you get an after image that's yellowish. If you look at yellow, you get blue, and so forth. So if you look at this for a while, and then look at this, the after image, the after effect is geared toward the lines, the tilt of the line. So if this one was red, which I think if this one or this one or this one was red, you end up seeing greenish after effects here. If this one was green, you see reddish in the other one. And so you might say, well, that's just perception. But it turns out that this effect lasts, if you look at it for a long period of time, for 5 minutes or something, it lasts for a day or 12 hours. It lasts for a long time. And there are other similar effects. So it could be that the tilt of these lines is the predictor of the color, I guess. The predictor of the color. And color is an opponent system, just like, I guess I've been arguing everything is. Color is an opponent system. If you want, if those of us who have done color photography know that if something is too bluish, too cyan, you have to add reddish. You have to add red, and that neutralizes it. So it looks to me like it's the same. Okay, that's it. Any questions?